Hello, everybody. This is Professor Louie. If you're watching this right now, you are probably enrolled in Spring 2018 Cognitive Neuroscience. I'm sorry I can't be there with you physically, but I'm very much there mentally. So, without further ado, let's dive right in. What is cognitive neuroscience? Cognitive neuroscience is fundamentally the study of how the brain enables the mind. So that includes the relationship between structure and function of the brain and how it gives rise to processes such as vision and audition and sensory processes, motoric processes, and then also cognitive processes that includes attention and memory and language and emotion and eventually social function and also development and evolution of the brain. Where is the mind and how do we measure it? We all know that the brain is a 3 kilogram mass of matter and it's sitting inside your skull and it's got about the consistency of Jello mixed with cream cheese. Okay, but somehow inside that 3 kilogram of matter lie all your thoughts and all your personalities and the way you relate to each other. So how does that come about? Now, a lot of people have asked this question, and it really goes all the way back to very early days of with philosophers, students of natural philosophy, and it goes all the way to modern neurologists, and also, of course, neuroscientists are kind of the main people looking at this right now. But then we also had quite an interesting history with the earlier behaviorists, right? People who used behavior to understand the brain, and then also cognitive scientists who tried to use cognitive models and psychological constructs to understand the contents of the mind. Now, early depictions of the brain so start a long time ago, but we'll start our discussion with Da Vinci, who started building models of the human brain around 1500 when he dissected human cadavers, and specifically the brains of human cadavers, and he realized that there were some areas of the brain that weren't just matter, right? There were actually some fluid-filled structures, also known as ventricles, in the brain. And so by injecting wax into these fluid-filled cavities or ventricles, he was able to build a model of those ventricles. And what he realized was that these ventricles were all interconnected in the central nervous system. Right, so from the brain all the way down the spinal cord, towards the body. And so he thought that because that was a more fluid part of the brain, he thought that was where the mind was, right? That was not necessarily, he didn't think it was the brain or the gray or white matter of the brain that was doing the work. He thought that the soul was actually in the ventricles. Now, of course, we know that that's not true, but that's one of the earliest attempts to map brain structure onto function. Fast forward some years, we had René Descartes, who was famous for his ideas about mind-body dualism. And so around 1647, he actually started to map out what he thought a mind-body relationship would look like, right? And so this is kind of Cartesian dualism, a map of Cartesian dualism. And so he thought that he looked at the brain, the human brain, and realized that one region that was asymmetrical, like the only region that he found to be asymmetrical, is in the pineal gland, which is a small structure that actually we know now to be regulating hormonal functions. But because he saw that it was in the middle, around the middle of the head, and relatively asymmetrical, he thought that must therefore be where the seat of the mind was, in the head. And the way he thought that the body connected to the mind was in this very simple circuit that he diagrammed. So you can see how, when you have a potentially painful stimulus such as a fire, right, you have some sensors within your body that then, as soon as they touch the fire, then it sends a signal, right, all the way up your spine into your soul, right, into your pineal gland, and then goes back down in order for you to withdraw your hand quickly, right? So this is a simple, what we now think of as a simple reflex arc that he mapped out as an example of this, of the way in which the mind could influence the body. Now, again, we know that this is not quite the story now, but it was a first attempt to take a circuit or systems level view of thinking about the mind-body problem. Now, then, in the 18th century, we had Franz Joseph Gall, who was himself a physician. He was a doctor, and he started realizing, he started noticing that humans have bumps on their scalp, right? And they're bumps on their skull. And he started formulating a theory which was derived from measuring these bumps on the skull. Now we know that this is quite ridiculous now. I mean, but this was the study of phrenology, which did gain a lot of traction back in the day. But this was the study of how these bumps on the skull might map on to certain cognitive or characteristics or individual differences in the mind. And so he thought, for instance, that this bump at the top of your head, right, maybe you guys can all kind of touch your own heads and see where the bumps are. He thought that the bump on the top of your head meant you had a firmness of purpose, right? So that you were a determined person. And then he also thought that the reproductive instinct, also known as amativeness, was mapped onto this bump that's close to the back of your head. So amativeness. Now you can maybe touch the back of the head of your two friends sitting close to you or something. But we now call these the mastoids. So these are just the bone structure at the back of your ear, right? So if you feel behind your ear, immediately behind the flappy part of your ear, you have these two bones that stick out a little bit more in some people than in others. And what Franz Joseph Gall thought was that if you had a more pronounced bump in your in these mastoids, that you were more amative. In other words, you were more interested in sexual reproduction, right? Or you're more interested in love. So you can decide which of your friends is more amative than the other. But of course, this is kind of a silly idea, but it did lead to ideas like criminal phrenology, right? Trying to measure people's bumps on their heads to see if they might map on to how suspicious people are, or maybe how criminal somebody might be, and also their individual differences in how likely they were to catch diseases and disorders like consumption, right, lung disorders, so general health. And then it also led to some ideas we know to be fundamentally false now of eugenics, right? So the idea of relating genetics to behavior, or one-to-one mapping between genetics and behavior, and then eventually breeding people to show a certain kind of certain kinds of more desirable characteristics. Now we know that this is quite untrue and also irresponsible and unethical now, but it did really, Franz Joseph Gall, right, for all we ridicule him about in modern neuroscience, we do agree that this idea of mapping structure to function does stay on as being an important idea in an important kind of guiding principle of cognitive neuroscience. Now, a much more recent and I think idea that's a bunch of ideas that's still much more accepted today was from Paul Broca, who was the 19th century French neurologist, who started taking another approach to the localization problem of localization of brain function. So just if anyone has been to Paris, I'm sure some of you have been to Paris here. If you go to the Eiffel Tower, if you approach it from the back, you could see that Broca's name is actually honored in letters in the back of the Eiffel Tower. Now, so what made Broca so famous? This is the person. This is the brain of the person, formerly known as ton. So in textbook it says tan, but it's actually French, so it's pronounced ton. Paul Broca was famous for seeing this patient who came in with a lesion in his brain, right? So he didn't have the techniques to find the lesion before the patient died, but he saw this patient who couldn't say anything except tong 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 tong. And then after the patient died, upon autopsy, he realized that there was a localized lesion, right? So there was death of brain matter in this region that we now know to be to call the inferior frontal gyrus. So Broca's, because Broca related this region to the function of language, right? So and thus in general, starting to find some evidence for individual parts of the brain that might be related to in specific cognitive functions such as language. He really started, in a way, the modern revolution for how we think about structure-function relationships of the brain. And so this region, known as the inferior frontal gyrus, anatomically, we also think of it as the Broca's area. This is the famous Broca's area. Now, in 1861, right, he found that this region was missing in his famous patient Tong. And since then, this is one of the things that have really held up, kind of findings that have held up over many, many years. Since then, we've had structural and functional magnetic resonance imaging technologies that have also pinpointed generally the same region, inferior frontal gyrus, that are involved, causally involved, in the production of speech, right? So because of this idea of a specific region of the brain being tied to specific functions, cognitive functions such as language, Broca is really credited to be the first to systematically shed light on the localization of brain function. Now, fast forward some more years, we had more evidence for the localization of brain function. And so this is Dr. John Harlow, who was an American physician, and he was famous for his patient Phineas Gage. Right? Some of you might have heard about Phineas Gage from a previous neuroscience course. And so Phineas Gage, we'll talk about him more in future lectures, but when he was 24, he was working in a railroad as a foreman. So he was kind of, I guess, supervising railroad work. And due to an unfortunate accident, this gigantic spike, that's about 3 feet long, a metal spike, got into an explosion, and the spike went right through his head. It went through his eye and out the other side of his head. This is a picture of his skull. I took this picture myself. It was in the Countway Library at the Harvard Medical School. It's next to a sign that says "No photography allowed." But hey, but you can quite clearly see that where the tamping iron had gone right through his skull. Right, so it went through the head and then out of went through the eye and then out of the skull. And what was interesting was that after this event happened, miraculously, Phineas Gage survived after the accident, but personality-wise, he was no longer Gage. His personality had changed from a rather amicable, nice young man to a very easily angry, irritable, and tough person to be around. So this is a three-dimensional reconstruction of his skull and how the trajectory of the tamping iron. And so you can see that by going through the eye and out the top of the skull, the tamping iron took out a small chunk of the brain tissue that we now know to be in the orbital frontal cortex. And we'll talk much more about the orbital frontal cortex throughout the course of cognitive neuroscience. So this is more about more evidence for how individual brain structures might map onto brain functions. But from another perspective, how does structure give rise to function? It's not just which structures give rise to which function, but how do these structures give rise to function? So another Canadian neurosurgeon, his name was Wilder Penfield. He was recently honored by a Google Doodle. Wilder Penfield started to do this very systematic work of mapping individual pieces of cortex in the brain to function. And he did so by taking electrical recordings during of human subjects during open brain surgery, human patients. When they were undergoing surgery, he would put an electrode on the somatosensory cortex, which is immediately behind the central sulcus of the brain. So it's a little bit, it's near the middle, a little bit behind the middle line. And we'll talk much more about the neuroanatomy in the next lecture. But he took electrodes and stimulated these individual parts of the somatosensory cortex. And he realized that when he was stimulating individual parts of the cortex, the patient reported feeling sensations on different parts of their body, right? So if he stimulated this part, this part of the cortex, the patient said, "Oh, I feel stimulation. I feel touched on my leg, right? Or I feel touched on my foot, or on my genitalia, and so on, or my hand." And what was interesting was that there was not a strict one-to-one mapping between the size of the body part and the size of the brain area that was corresponding to that body part. Right. So there were some parts that were more sensitive, tended to have more brain real estate kind of devoted to it, right? So hands and fingers, for instance, are very sensitive parts of the body, and so they mapped onto larger regions of the somatosensory cortex. And so this is a reconstruction of a one-to-one mapping between the size of the body part and the size of the brain region that was devoted to the sensation of that body part. And so this is known as the somatosensory homunculus. A homunculus just means a little man. It's a small person who was whose body parts are proportional to the amount of neural real estate that are dedicated to that body part. And so you can see that the more sensitive parts of the body tend to be larger, right? So our lips are quite sensitive, right? Our fingers are quite sensitive. Torso is not so sensitive, right? So this is rather funny-looking person is the somatosensory homunculus, which represents the amount of neural real estate dedicated in the somatosensory cortex. So that's a really elegant way to relate the structure of the brain to individual specific functions. Now, drilling down a little bit more into individual pieces of brain tissue now, so people have started to ask, then how exactly do the cells of the brain function together? And so this goes back into the question of what do neurons look like and how do they work together? And so there were two influential scientists, Camilo Golgi and Santiago Ramon y Cajal. One was Italian, the other was Spanish, and Golgi was really quite famous at the time for his reticular theory, and so that's the idea that individual cells of the brain work together as one continuous sheet, right? And so reticular just means layers, and so he thought that the neurons were all connected together in one gigantic layer. Whereas in contrast, Ramon y Cajal espoused the neuron doctrine, which we now espouse, right? So he ultimately won the debate. But what he thought was that the brain, the fundamental structure of the brain, is in individual cells, a large number of small neurons or brain cells that connect to each other, right? That are talking to each other. And so they ended up sharing the Nobel Prize for Physiology and Medicine in 1906, and they kind of together founded this idea about fundamental structure of the brain, and so they both arrived at their conclusions by doing staining studies, and so these are very careful studies of microscope studies that looked at brain tissue, and then they both made drawings of of how individual neurons functioned. Individual neurons were wired together, and so these are some examples of drawings by Ramon y Cajal, and you can see how he, depending on the kind of stain, right, you can use different stains to show more the connections between the different cells and also the individual cells themselves. Right, so by refining the staining techniques of Golgi, Ramon y Cajal eventually showed that the cells are individual neurons themselves, and that they function together not as one continuous sheet, but individual points that connect to each other functionally. And so that's now known as the neuron doctrine, and for that we pretty much think of Ramon Cajal as a father of modern neuroscience. But the neuron doctrine states that neuron is the fundamental building block of the brain, right? And that the connections between neurons are fundamental to how the brain communicates, and that then gives rise to behavior. Right? So again, individual neurons, each with their own structures and with their own biological functions, actually act together to enable more complex behavior. So going back to our origins of cognitive neuroscience, where I've started talking a little bit about philosophers, and then moved on to talk about neurologists and some neuroscientists, and some biologists, and now I'm going to talk about some perspectives from psychology. I'm starting with behaviorism. Right, so Skinner, B.F. Skinner, was an American psychologist in the early 20th century, who really started to think about behavior as a systematic relationship between stimulus and a response. Right? So he was famous for coming up with this experimental setup. It's known as the Skinner box, and it's useful for operant conditioning, which we'll talk much more about in the later days of this course. But what that does is it's actually quite simple. It's just a box where you can put an animal in, right? An animal such as a rat, and you can give it some stimuli, right, such as signal lights or some speaker through which you can deliver sounds, and in response to certain behaviors on the part of the rat, you can give the rat rewards, such as a pellet, a food pellet, or a punishment, such as an electrical shock. Right? So by simply giving the rat certain stimuli, you can observe and control its response. Right? So that's the fundamental tenet of behaviorism, is that the brain, right, or it doesn't actually matter what's inside the brain, it's just you can treat it as a black box, right, and it's just a systematic mapping between stimulus and response relationships. Now, fast forward some years, this idea gradually fell out of favor, so behaviorism gradually gave way to the cognitive revolution, and one of many kind of important figures of the cognitive revolution was Edward Tolman, who's an American psychologist working out of Berkeley. There's a Tolman Hall now in Berkeley, and so how he arrived at the idea that it's not true that this black box doesn't matter. What's inside the black box actually matters a lot, and he arrived at these conclusions with quite a famous Tolman maze. Right? So this is a maze. I show you this. This is simply a 4-arm maze, right? And this is a maze for rats. And what he could do is he could show a couple of things. Right? You can put a reward, right, such as a food pellet, on one arm of the maze, and then you could place the rat in a different arm, and you can see how long it takes for the rat to arrive at the food reward, right? And you can just measure the number of errors that they make, and what he observed was that the more hungry the rat was, the sooner it learned to find its reward. Right? So the state of the being does matter. And what he also found, and this is really quite clever, is that the rats seem to have a mental, a cognitive map within their brains, right? So how he found this was he would put the rat in A, or spot A, for instance, um, and he would put some food in B, right? So the rat, over several trials, learned to run to the middle and then turn right, right? So the rule was just to turn right. Now, then he would remove the removable barrier, which is coming out of C, and now place the rat in arm C. Now, if the rat had just learned to turn right, it should go to D. But instead, it went to B, right? So it's not just that the rat is learning a simple turn right rule, it is as if the rat has a mental map of the overall maze inside its head, right? So this is a really powerful method, and simple but powerful method that really showed that we're not just simple rule learning machines that relate stimulus to response, but that the state of the organism and the contents of the mind do make a difference, right? And you can actually store some more complex representations, such as maps, in the mind. So it's not just a simple black box relationship between stimulus and response, but the organism itself actually matters, and there are actually individual psychological constructs, right? So that's actually what these little blobs are supposed to represent. They're constructs, or kind of core pieces of information that are inside the black box that actually make a difference for how the stimulus translates to response. So going back to our summary of our behaviorism and cognitive science, we're starting to move towards using cognitive models to understand the psychological constructs that are mediating the relationship between stimulus and response, in contrast to the classic behaviorist view, which treats the brain as a sort of black box. And I'll close by this idea that modern cognitive neuroscience is really a confluence, or again, a Venn diagram between neuroscience and psychology, but also neurology and cognitive science. And over the next few days, we'll talk about a variety of neuroscience techniques. And I'll just mention this really important point, that these different techniques that we're going to cover in the next couple of weeks really do converge and complement each other. And so this here is a map of structure and function. So structure being the resolution, the spatial resolution with which you can image, or you can find information about the brain, and function being the temporal resolution through which you can resolve different time, the brain activity over different time differences. Now, and each of these areas here represents one method or technique through which you can understand the brain, right, from lesion studies to patch clamp recording to single cell recording, and so on. And so each of these occupies a certain space in your map of spatial temporal resolution. We'll come back to talk a lot about this, but the point right now is that we'll talk about lots of different neuroscience techniques that are supposed to converge and complement each other. And another way through which we can accomplish our a kind of more nuanced understanding of the brain is through meta-analysis, right? So it's not just by doing one experiment at a time, but by more kind of conglomerate, aggregate understanding of a set of different studies all at once. We can start to understand how not just individual areas of the brain give rise to individual functions, but how groups of areas in the brain give rise to groups of functions. And I'll just end with this small puzzle here. You can see right here that this human subject is undergoing some kind of procedure. She's having this put over her head, and the question for you is, what imaging device is this? What is this imaging device? Okay, so I think I will end on that. I'll end on that cliffhanger, and I will see you next time.